Hello, welcome back to the General Microbiology. This is practical session lecture 8, fifth semester, College of Pharmacy. Our topic today is the smearing and the spore stain method. So if you remember, just to remind you, I would like just to give you some a little remind about spores. When in theory in general microbiology, just to remind you that endospore, if you remember, please. So endospore, we have the principle that some bacteria can produce spores. So not all of bacteria, of course. And why they do this? Why they produce the endospore? It is type of the reproduction, of course. Type of the reproduction. For example, Bacillus species and the Clostridium species. This is just for an example. They produce endospore. It is like a complex developmental cycle that produce resting stages, call it this endospore. So as a definition, we can give this look, please. It is a dormant form of the bacterium that allows it to survive itself from the poor and the bad environmental conditions. This is as a definition, please. And what are the main characteristics of endospore? So the endospore, we say that it is a dormant form that to survive the bacteria from the bad environmental conditions. So it is, of course, this there is no nutrients, lack of nutrients, the bad conditions in general, like temperature and others. So in these cases, in these conditions, which are the not favorable for growth, they produce endospore. They state itself in the cycle of a sporing. And this, after the conditions come back, I mean the good conditions come back, they will again germinate. They will germinate and do the germination to put in the vegetative cells, of course. So here, if you know that endospore, it's a dehydrated structure, and this structure resistant to the bad conditions, bad environmental condition, of course, like heat. I mean, high temperature, high heat, radiation, and acids, and many chemicals as well. So these cannot kill the spores, or the spores cannot killed by those conditions. I mean, bad conditions. Of course, they have levels. For example, not for a very high temperature. It can keep itself. It can stay and keep itself to a good level of a high level of heat and radiation, of course. We talked about what are the main characteristic. So the main is resistant to the this high heat and acids and radiation and chemicals as well. How they do this? Why they do resistant, and how? What it has characteristic to protect itself. So they have a protein code called exosporium. They have a protein code called exosporium. This exosporium can I mean, it is made of protein keratin, called exosporium, which made of the protein keratin that forms a protective barrier from the and this protective barrier resists the heat and other conditions, of course. Heat resistant is very associated to the content of the water content. I mean, water content of the endospore. As much as the higher the water content of an endospore, the less heat resistant to the endospore will be. I mean, as much the water content in endospore is high, it will be effect on the resistant to the heat. Of course, the less heat resistant to the endospore will be. This is the association between heat resistant and the water content of the endospore. For example, if you have any question you may expect the question that why and how they do I mean why and how they do resistant to the bad conditions, and and what the association or relationship between heat resistant and the water content. Look, this is the and slides under microscope. You're going to find like this. Why? Which is the red and which is the green? We're going to talk about it in the procedure, of course, and in the test today. Another slide, please. Look, if you come back to the slides, the bacterium cell contains the main I mean contain the two contain the cell, the coat, and the spores. The coat, the protein coat, and the spores. So the spores will stained by a specific color, and the cell will stain it by specific color, of course. Look here, it is red and green. The green is the color of a spores, stained of spores, and this the red is still the protein, the cell around the spores. Here it is like crystal violet or sorry, the other stained. Look, the spores is blue or light blue, and the cell is red. So the test today, we're going to do staining by malachite green. Malachite green or malachite green, they will penetrate the endospore and colored the spores by the green color. I mean, when you stayed, when you after smearing, we're going to talk about it in the procedure, of course. In when we do the smearing fixation and put the malachite green for 5 minutes, 6 minutes, and then in this cases on the water bath, of course, I'm going to show you, and you will work in your hand. The malachite green will penetrate to the endospore and becomes entrapped in the endospore. And this malachite green not removed later by washing or decolorizing agent or water, will not removed by washing or decolorizing agent or water. They will stay in green. And the other, the cell will keep the red cell. I mean, they will not keep the malachite green, and will have the last stained the last strand will which will stated it, which is safranin in our test. So in our test, look, in our test we have three materials, of course: malachite green and safranin and water or decolorizing solution. Three solutions are important in our method, of course. So the name of our method is Scaffer-Fulton method by malachite green. To stain the endospore, and safranin will stain the vegetative portion of the cell. As I said, the protein coat of the cell. At first, you will have a slide and do the smearing from the method. At first, you're going to have a slide and have the the culture, a small part of colony from the culture, and put on the slide. Then do the heat fixation or fixed after they dried, and follow the steps of the staining, of course. This steps is very easy. So of course, they will. So in this method, our example is Bacillus spp, the one species of the Bacillus, which can produce the endospore. At first, you're going to have a slide and do the fixation after dried, heat fixation, I mean, on the beaker, on the water bath. Then follow the steps of the staining, of course. I'm going to show you now in the procedure, of course. Of course, after you do staining under microscope, you may find the spores, the located of the spores in the middle, which called is central, or in the end of the cell, which is called terminal. Central or terminal, or maybe between the end and the middle, which is subterminal, of course. This is the location of the spores, of course. A simple image about the stain. So cells will stain it like before the malachite green. After staining, the malachite green will cover all of the cell by the green color, and then in the here in the decolorizing stages or steps by water or decolorizing solution, the spores will stay in green, and the other will remove the color will remove by in the protein coat. Finally, the cell, after we do the safranin will stain the cells, and malachite green in the spores. Materials, as I said, is the sample, Bacillus culture, and the malachite green stain, safranin, and staining rocks, and of course cloth stains. Procedure is very easy. As I said, is prepare a smear of organism, and as you do smearing in the last lecture. And then the fixation by heat on the water bath, or sometimes we can do by hand on the above of the Benson burner for some minutes, and then immediately you're going to smear the malachite and wait for 5 to 6 minutes. Green will penetrate, malachite will penetrate to the spores, And then after that, you're going to do washing by decolorizing solution or water. Gentle, of course, streaming of gentle stream of water, and then stain after that by safranin for 45 seconds. Uh, After that, you're going to do washing, of course. A green spores will be green, and the vegetative cell will be red. This is the steps and simple steps of the spore stain. Again, this is the smearing. You look, smearing and heat fixation. Malachite green, and this malachite green, as I said, 5 to 6, sometimes to 10 minutes. And the staining then by safranin. And after safranin, you're going to do the wash by water, and then it dried by filter paper or a towel, a piece of towel, and then put the oil drop and under microscope by the oil lens to watch and to see the spore stain and the cell, of course. Malachite green, I mean, the spore will be green and the cell will be red. Look to this. It is better to do the fixation by this shape. I mean, look, it is water bath, and put the slides here for with malachite green from 5 to 10 minutes, and then washing, and then safranin for 45 seconds, or sometimes it is 30 to 40 seconds, and then washing again by water, and then dried by a piece of towel or water filter paper, and then examined by oil immersion under oil lens to say that. Thank you for your listening. This is the last of our lecture, and if you have any question, of course, you can ask me. And in the lab, you're gonna to work in your hand, and feel free to ask any question or to additional information, of course. Thank you for your listening.